2020 జేఈఈ మెయిన్ ఫలితాలలో NSR ఇంపల్స్ విద్యార్థుల ప్రభంజనం. ఆల్ ఇండియా అన్ని కేటగిరీస్లు 18, 35, 251, 253, 582, 606, 778, 780, 812, 821, 1031, 1039, 1303, 1575, 1654, 1753, 1754, 1978 ర్యాంకులే కాక మరెన్నో ఉత్తమ ఫలితాలు సాధించిన విద్యా సంస్థ NSR ఇంపల్స్.
A bunch of flagella. Bacteria with a tuft of flagella on both ends. Means both terminal ends they contain a tuft of flagella. So here now if you look at, this is how the bacteria looks like. So here this is the bacteria. Now you can see this, there are so many flagella at one corner. Similarly on the other end also we are drawing so many flagella. Tuft of flagella, group or bunch of flagella on either side of bacteria. We can see on either side of bacteria. Such type of arrangement is called as Amphitrichus bacteria. Amphi means both sides, both terminal ends, both corners you can see a tuft or bunch of flagella on either side of bacteria. Such type of arrangement is called as Amphitrichus. Let us take some examples for Amphitrichus. Nitrosomonas, Nitrobacter. What are the examples? Nitrosomonas, Nitrobacter, which converts atmospheric nitrogen to, to ammonia, into ammonia. Where this bacteria plays an important role? This Nitromonas nitrobacter converts atmospheric nitrogen to ammonia. Then such type it's called as what? Amphitrichus bacteria. Means bacteria with a tuft of flagella on both ends. Here also we can see, here also we can see, right? Example, Nitrosomonas nitrobacter, which converts atmospheric nitrogen to ammonia. Such type of bacteria are called Amphitrichus bacteria. Let's have one more category. So let us take the next category as Peritrichus bacteria. Let us take a Peritrichus bacteria. What is meant by Peritrichus here? Here bacteria or flagella occurs all over the surface of bacteria. What is meant by Peritrichus? It means that flagella occurs all over the surface of bacteria. Such type, if you look at how it will be, if this is a bacteria, let us take this is a bacteria. Now in this case flagella occurs all over the surface like this. Flagella occurs all over the surface. Now on the total surface around the cell wall you can see flagella originating at different corners in different directions. The flagella have bacteria has flagella on the total surface of bacteria. Such type it's called to be said to be as what? Peritrichus. Now let us take example for Peritrichus. So you can take E. coli as well as Salmonella. Okay, so what are the two examples we have to remember? E. coli and other one is Salmonella. What is E. coli? E. coli it's called as Escherichia coli. Escherichia coli. What's the meaning of this? Escherichia is a genus name and coli it's a species name. Right? What is the bacteria? E. coli. Other one is Salmonella. Salmonella typhimurium. Like that we have different categories. E. coli meaning Escherichia coli. Escherichia is nothing but a genus name. Coli is nothing but what? A species name. Salmonella typhimurium which causes the disease typhoid. But here we have some important point to remember about E. coli. Now let us see that here. But before that try to remember peritrichous meaning here flagella occurs all over the surface of bacteria. So here flagella occurs from all the corners. This is how the bacteria looks like and you can take example E. coli. E means Escherichia. Coli. Escherichia is a genus name. Coli is nothing but species name and also we find another example Salmonella. E. coli, Salmonella both are nothing but examples for the category called as peritrichous bacteria. So here let us take some note points. Note points means some important points to remember, means the question getting points, questions means from these statements there is a possibility of getting the questions. They are said to be as what? Note points means most important points which we get in the form of a questions. Now if you take E. coli is peritrichous, Salmonella is a peritrichous. Now if you look at, in this case if you look at, let us talk about an important statement about E. coli. E. coli is a common inhabitant of E. coli is a common inhabitant of human intestine. So E. coli will take shelter from the intestine. So in the human intestine most commonly found bacteria is nothing but Escherichia, that's called E. coli. So E. coli it is common inhabitant means it most commonly takes the shelter from human intestine. Of course it has some important role, it helps in checking out the pathogens, it helps in digestion of the food. So it is a favorable bacteria for the humans and most commonly found in case of human intestine. So Escherichia if you look at that has the condition called as peritrichous. This point is very important and it was the previous NEET question. Previously we have got this question saying that which bacteria is a common inhabitant of human intestine, which bacteria most commonly takes shelter from human intestine. At the time you need to take the option called as E. coli. E. coli is found in our own intestine, right? It's a previous NEET question, very important, repeated quite good number of times. So remember this point, right? We will discuss even in second year also, you don't have to worry, right? Now apart from this peritrichous, another example is nothing but Salmonella. Now if you look at Salmonella, generally Salmonella is just genus name. Typhimurium, it's a species name. means we can take that Salmonella typhimurium. Salmonella, it's a genus name. Typhimurium, it's a species name. Salmonella typhimurium causes the disease called as typhoid. Remember, these two will come under the category called as a Peritrichus bacteria. Again I say, even classification of bacteria based on flagella is also important. What is the first category we say? Atrichus means bacteria without flagella, atrichus. So try to remember the examples are very important. Lactobacillus pachyderella. Next one is Monotrichus, bacteria with one flagella. Monotrichus, you can take example Vibrio xanthomonas. Lophotrichus, a single or tuft of flagella present at one corner, it is called Lophotrichus. Example, what you will get? Spirillum. Then Amphitrichus means tuft of flagella present on either side of the bacteria. You can remember examples like Nitrosomonas, Nitrosococcus, Nitrobacter. There are so many different bacteria. Mostly these bacteria involved in conversion of atmospheric nitrogen into ammonia. This point we need to stress even in our second year syllabus. At the time we will discuss the role of these bacteria and how they involved in conversion of atmospheric nitrogen into ammonia, that we will discuss in the form of nitrogen cycle. At the time I will ask you, Nitrosomonas contain what type of flagella? So type of flagella is a first year concept. How they play a vital role in nitrogen cycle is a second year concept. So when I ask you at the time of second year, Nitrosomonas contain what type of flagella? By the time in your second year, you should remember the first year today's concept. You have to tell me, yes, they are examples for Amphitrichus. If you remember that extent each and every point, that's an awesome preparation for the final NEET. Now apart from this, some contains Peritrichus, where flagella occurs all over the surface of the bacteria. You can take example E. coli, Salmonella. Remember in bacteria also we have two names. One represents genus name, other represents the species name. Of course, species name we have to start with small letters. Right, Escherichia coli, Salmonella. On these two bacteria I have given some important note points to remember. E. coli is a common inhabitant of human intestine. E. coli most commonly we found in the human intestine. And Salmonella, it was a previous NEET question, try to remember. And Salmonella typhimurium, it was responsible for causing the disease called as typhoid. In this Salmonella genus has so many different type of species, but among them one of the species typhimurium alone responsible for causing the disease called as typhoid. Now this is nothing but classification of bacteria based on arrangement of flagella. Flagella helps in locomotion, one place to other place. Now let us continue our discussion here. Now here the bacteria, let us differentiate prokaryote as well as eukaryote with one point. Right, let us differentiate with one point. What is that one point? Flagella. Flagella is lack of 9+2 arrangement. In case of eukaryotes, we say flagella possess 9+2 arrangement. Better you can write contain. Flagella contain 9+2 arrangement, or possess, you can write anything, you can write, no problem. So this is a very important point you have to remember, a difference in terms of flagella. Right. So flagella is present in case of both prokaryotes as well as eukaryotes, no doubt. But in case of prokaryotes, the arrangement of flagella is different from the arrangement of flagella what we find in case of eukaryotes. How the arrangement differs here? Flagella is present in prokaryotes, but here it is lack of 9+2 arrangement. Okay, whereas in eukaryotes, flagella contains a 9+2 arrangement. So, what is this 9+2 arrangement which is absent in prokaryotic flagella but present in eukaryotic flagella? The 9+2 arrangement of flagella we are going to discuss in detail in the coming chapter called cytology. At that time we will draw the diagram and we will come to know what is the arrangement of flagella which we find in case of eukaryotes when compared with the prokaryotes. Now presently for your understanding I will just draw the simple diagram. Now in case of eukaryotes, if you look at, so here we find 9 like this microfibrils we draw generally. Let us draw somewhat big, then only you can understand. So here it is like this. These are called microfibrils. The flagella is made up of microfibrils. What are these microfibrils? They are nothing but proteinaceous extensions what we find in the cytoplasm. So these all occurs in pairs, 2 like this. It's a pair containing how many? Here they occur in a pair. Pair means how many in number? 2. Like that, how many are present? They all are interconnected by linkages, or they are called intermicrotubular bridges. How many in number? From here if you start counting, 1, 2, 3, 4, 5, 6, 7, 8, 9. So there are 9 pairs, each pair contains 2. Right? But 2 has one more meaning. Now if you look at in the center, you find more 2 individual microtubules which are connected with the center linkage or intermicrotubular bridges. Right? Rest of the labeling part we will discuss at the time of cytology, the coming chapter. Just for an idea, I said this to remember, because here you have to remember only one point, that is prokaryotic flagella is lack of 9+2 arrangement. That means 9+2 arrangement is totally absent, whereas eukaryotes has flagella but they show 9+2 arrangement. This one point is very important to remember in this chapter. But what is 9+2 arrangement? It is a big question mark to your mind. That we will discuss at the time of cytology chapter, where we draw 9 pairs of microtubules. Center more 2 are there. So we call this arrangement as 9+2 arrangement. What are these 9 here? So here flagella is made up of microfibrils. 9 pairs of microfibrils are arranged at the peripheral, and 2 individual microfibrils are placed exactly in the center. That's why the arrangement is 9 pairs plus 2 individual, 2 singlets in the center. The arrangement is called 9+2 arrangement. The labeling part, what are these bridges, what are these linkages, what are these microfibrils, what is there in the center, total labeling part, and in detail study of flagella we will study once again very clearly in depth at the time of cytology in the next chapter. Okay, so this is very important point. Prokaryotic flagella lack of 9+2 arrangement, and prokaryotic flagella possess 9+2 arrangement. So wherever we find differences between prokaryotes and eukaryotes, you have to remember very clearly. Hello students, now let us continue with the discussion about biological classification. Now where exactly we are? We are in the discussion about bacteria. Now when we take the bacteria here, now let us say that bacteria has the cell wall. So here outermost layer it's called as cell wall. Just inner to this what we find it's called as cell membrane. Outer layer what we find it's called cell wall, inner to this what we find it's called as cell membrane. But here the outermost layer of the bacteria, generally we call that layer as Glycocalyx. Generally outer membrane of bacteria, generally we call it as what? Glycocalyx. Guys, whatever I write on the board with a clear handwriting, I hope it's very clearly visible to you. With the clear handwriting, make sure that you write your notes, running notes very clearly, and that day when you can read out all the points, whatever we discuss in the class, if you just can have a look at on the diagrams as well as the classification as well as the block diagrams, what I am trying to explain here, very neatly if you can write and maintain your own running notes, that becomes very easy for you to understand the concept, and it's easy for you to make sure that it is easy that you can remember the topic and you can learn the topic in very short time, and you can remember for longer duration of time, provided if you complete the preparation on the same day itself. You are listening to the class, write down your running notes and revise once your running notes, write without saying on the paper. When you can do this little effort on each and every class, guys, then your preparation becomes very easy. Chakkaga running notes raskoni chakkaga prepare. You just have to recite yourself orally or just you write once on the paper. Then you will remember forever, because at the time of second year, these all topics are interconnected. If you make sure that your basics of first year are very strong, then definitely you can follow second year classes very well, right? So here outermost layer, what you find in the bacteria itself is called Glycocalyx. So what I am trying to explain here, what is Glycocalyx here? It's nothing but an outermost layer of bacteria. Outermost layer of bacteria. So always I am just drawing the empty box. It's not actually empty box. You know that it has its own nucleoid, it has genophore, it has ribosomes, and here it has its own cytoplasm. So all the time I am not drawing the inner content of bacteria, but whenever you draw, make sure that you will draw the inner content of bacteria, right? And just don't make empty box, right, as I am drawing. Now if you look at in this case here, what is Glycocalyx means? It is a outermost layer of bacteria. It's a single bounded membrane, but it contains, it looks like a single membrane, but it contains three layers. Actually it contains a combination of three layers. It contains a combination of three layers. What the three layers here? Now outermost layer, it's called as, the outermost layer itself is called Glycocalyx. This Glycocalyx may be thick and very tough layer. Then if the Glycocalyx exists as thick and tough layer, it's called as capsule. It's called as what? A capsule. If the Glycocalyx, if it exists as thick and tough layer, it's called as what? Capsule. Or sometimes in some bacteria, it may not form the thick layer. It may exist as, Glycocalyx exists as a loose sheath or a slime layer. Loose sheath or a slime layer. It's called as slime layer or S layer. Slime layer or S layer. Okay, so when you look at here, Glycocalyx exists as thick and tough layer, which is called as a capsule, or sometimes it exists as a loose sheath, very loose, tiny, thin sheath, which is very slimy to touch. Slippery, slime means slippery. So the slime layer or the loose sheath what we find here itself is called as a slime layer, or we call S layer. Now in this case, if you look at, sometimes as I told you here, it may exist this Glycocalyx as a thick layer, then we call this as capsule. So, I am drawing thick layer here because sometimes we find a thick layer. Now, this is what is called capsule. And what is the other condition, as I told you to remember? Sometimes the bacteria contains, I am drawing the smaller one here, it may contain a slime layer, a loose sheet. This itself is called as S layer or slime layer. Some bacteria contains slime layer or S layer, and or some bacteria contains thick wall called capsule. Whenever capsule is there here, capsule protects the bacteria from extreme environmental conditions. So, what is the function of capsule? Capsule protects bacteria from extreme environmental conditions. Extreme environmental conditions. So, the bacteria is protected safely from, even if it is kept in high acidic condition, high salt condition, some bacteria has ability to tolerate. Why? Because they contain capsule. So, what is the point to remember here? Capsule protects the bacteria from extreme environmental conditions. Right? Archaebacteria has capsule. In last few classes we studied about archaebacteria. Archaebacteria has the capsule, protects the from extreme environmental conditions. And the next layer in this, so total layer itself is called Glycocalyx. In that outer layer itself is called Glycocalyx. Again, now then below this what we find is called cell wall, and below this what we find it's called cell membrane. So, here cell wall protects the bacteria, from bursting. Cell wall protects the bacteria from bursting. Bursting means whenever bacteria take more nutrients, more water, it will swell up in size, but still bacteria will not burst, it will not get destroyed because cell wall is flexible and it protects the bacteria from bursting or collapsing. And as usual, inner layer, cell membrane, it is nothing but inner layer which is a living layer. Inner layer or inner membrane, it shows some invaginations, and those invaginations are called as what? Mesosomes. Right? I think I told you for the last few classes, cell membrane shows invaginations, and these invaginations are called as what? Cell membrane shows invaginations towards inner side. These invaginations are called as mesosomes. So, you all know it very well. Mesosomes helps in respiration. Mesosomes, they play an important role in respiration. Not only that, the formation of the cell wall, septa at the time of division, distribution of daughter cells, distribution of DNA replication, separation of daughter cells, these all are nothing but the functions of mesosomes. Once again we are going to discuss about mesosomes at the time of cytology. So, in cytology we can discuss about mesosomes, right. Now, why I am telling about Glycocalyx? It contains 3 sublayers called, outermost itself is called Glycocalyx, below that we find cell wall, inner to this what we find is cell membrane. Now, let us mainly concentrate on cell wall. Let us concentrate on cell wall. Based on cell wall, the bacteria is classified into 2 categories, gram positive and gram negative. Now, let us continue discussion about cell wall. Cell wall is the second most layer, protects the bacteria from bursting and collapsing. So, let us discuss about cell wall. So, here now if we talk about the cell wall, you can take classification of bacteria based on the nature of cell wall. Better you can write your clearly the headings so that you can remember the concept very easily. So we can classify bacteria even based on cell wall. Classification of bacteria based on cell wall. Now let us discuss about this. Now when you look at the cell wall here, there is a scientist called as Christian Graham. It was given by the scientist Christian Graham. Now what Christian Graham has done it? Now Christian Graham has done a staining for the bacteria. Now when he has given stain to the bacteria, some of the bacteria could able to take the stain, they could able to absorb the stain, and some other bacteria they could not take the stain. So here Christian Graham is the one, he has applied the process called giving staining to the bacteria. So he has developed the method called a staining method. Right? So what he has done in the experiment, while doing the experiment, now he has taken two culture. So the one culture having the bacteria, this is a nutrient, this is culture plate. Culture plate itself is also called as Petri plate. A round small plate, glass plate will be there. In the lab you can see in your higher classes. Now there what is added, it's called nutrient media. In the culture plate we add nutrient media, meaning we add nutrients for the growth of microorganisms, especially for bacteria. Culture plate is a small glass plate with the closed lid which will be there. In that we are adding nutrients for the growth of bacteria. Now then what he has done is he has applied junction crystal violet. Let us take junction junction crystal violet. It's nothing but a dye. Itself we call it as a stain. He added a junction crystal violet, a dye or the stain is being applied onto the Petri plate here. When a dye or stain is applied and he has given time for the bacteria to take up the stain to absorb the dye. So after 24 hours, when these bacteria are observed under microscope, it is observed that some bacteria are taken the junction violet and appeared a dark bluish in color. So they appeared what is called as violet color. So some bacteria appeared in violet color. So here some of the bacteria, they are stained with violet color. But it is not uniform. All bacteria could not able to take the stain. What he observed is some bacteria are found to be colorless. Some bacteria are colorless. Only some bacteria could able to take the junction crystal violet stain. They absorbed the stain, they have taken the dye and appeared in purple color, or we say violet color. Either you can write purple color or you can write a violet color. Right? So some of the bacteria could able to take the crystal violet stain and they appeared in violet or purple color, and some other bacteria they appeared as colorless. So in the next day, then he did not get that why some bacteria has ability to take up the stain, why not other bacteria has ability to take up the stain, he could not able to understand. To get the clarity, the colorless bacteria which he removed and placed in another Petri plate. So I am just writing here. So he took another Petri plate, in that the colorless bacteria are placed. So these are nothing but colorless bacteria are collected. Right, with the help of a brush, the colorless bacteria are collected and put it in another Petri plate, culture media, and poured with nutrient media. The colorless bacteria what he collected with the help of a brush from this and kept here in the Petri plate. Again nutrients are added, then the bacteria started growing in and multiplied in their number. Now this time in the second step he added safranin. It's a pink color stain, a pink color stain. So all the bacteria became pink color. So all bacteria, let us imagine black itself as pink. So this all bacteria started stained with pink color. They could able to absorb the safranin and they appeared finally in the pink color. So that means some bacteria has ability to absorb junction crystal violet stain, they become violet color. The remaining bacteria don't have ability to take up the stain, but they have ability to absorb other dye, other stain called safranin. They could able to absorb the safranin and finally they appeared in pinkish in color. So then he started looking for why the bacteria don't have ability to absorb crystal violet and the same bacteria have wonderful ability to absorb safranin. Why some bacteria absorbed violet color and appeared violet color bacteria, and why not other bacteria take up the stain but they have ability to take up the safranin. That means some looks purple or violet in color and some bacteria looked pinkish in color. It's because of variations in the absorption of dye. That means there is a variation in the bacteria. Where there is a variation, finally with the help of experiment he could able to identify the variation is occurring in the nature of cell wall. Where the variation, the bacteria, some bacteria could able to take junction violet dye and some can take safranin. The variation is occurring, it's because of the nature of their cell wall. Now based upon the nature of cell wall, he has given the classification of bacteria. Now according to Christian Gram, the bacteria is classified into two categories, gram positive bacteria and gram negative bacteria. Again I repeat, according to the scientist Christian Gram experiment, it's called gram staining. The experiment what he has done itself, the experiment name itself is called gram staining. Itself is called gram staining. Now Christian Gram with the help of gram staining, he divided the bacteria into two categories, gram positive bacteria and other one is gram negative bacteria. Okay, he classified the bacteria into two categories based upon gram staining. Means Christian Gram staining, the staining is absorption of dye. Staining method, with that he could able to divide the bacteria into two categories. What are they? Gram positive. Which are gram positive? The bacteria which absorbed violet color. This one are called gram positive bacteria because they absorb violet color or purple color, appeared bluish in color. They have ability to absorb junction crystal violet. They are called gram positive bacteria. That means whenever you have applied two stains, the bacteria which appears violet or purple color stain, they are generally called as gram positive bacteria. And the bacteria which absorb safranin and appeared pink in color, generally these bacteria are called gram negative bacteria. So when you take the culture, when they get exposed to two different stains, if some bacteria appears purple or violet, they are called gram positive. Some other bacteria becomes colorless and after applying safranin they appear pink in color, they are called as gram negative. So try to remember, violet purple color bacteria are generally called gram positive, pink color bacteria are generally called as gram negative. Now let us continue our discussion here.